Hello. I'm always going to do that Seinfeld joke. Hello. This is DNA Workshop 2, and I am glad to have everyone to join me today. Last week in Workshop 1, we learned how to, load, explore, visualize mutation data, put them in the different formats, like the math format. And we did a little visualization like oncoplots, lollipop plots, things like that. And now it's time to go a little step further, where we want to discover statistically meaningful patterns and connect these mutations actually to real clinical outcomes. So why will be the difference in this workshop and the first one? Remember the first one we were like, what mutations exist? Just descriptively. Well, now we want to know which are actually significant, right? How many mutations per sample were they? Well, that's great, but do they actually predict something like survival? And where are they located? Well, okay, we have the position, but actually what pathways are they disrupting, right? And we want to do a little bit more visualization and connect this to, clinical correlation. So the model we're going to be using today is the pancreatic adenocarcinoma TCGA set. It's a bulk cancer set. And it's very interesting because it is so difficult to treat because of this lack of response to immunotherapy. And one thing it has is you'll see as near universal KRAS mutations almost in every single sample for the most part, about 90% on average, as clear molecular subtypes. It's very aggressive. And to learn more about some of these, hypermutation phenotypes and other issues with this cancer, we might be able to apply better therapy. So it's a great cancer to study because really it's just, if you see here, the survival rate's so low. So it's one of the most studied cancers to try to get some, at least some treatments that actually work, right? What are some genes we will counter? Well, KRAS, we mentioned, will be in pretty much all of them, right? Also, TP53 is a tumor suppressor that you well know, right? It's going to cause a lot of cell cycle disruption. We're going to see that early on, probably. CDK2A, SMOD4, and BRCA1/2. So as we move through this workshop today, what we're going to look for these genes and how they affect us, right? So we kind of talked about our objectives and what we're going to do here. We want to look at which genes are significantly mutated above the background. Do they mutually are they co-occur? Are they exclusive? Whether these mutations actually can predict survival, which pathways are actually disrupted, and which mutations are actually clinically actionable. So the first thing we want to do, right, is set up our environment. It's going to be pretty much a lot of the same packages that you have seen before. If you didn't do the DNDSCV from last week, there's the dev tools kind of talk install, and here kind of all our packages loaded with the annotations, and I'll let you in your own time to kind of go over those and see what is being used for today's workshop. And so first what we're going to do is load that TCGA pancreatic cancer data set. We have our mutation data, same query as before. All I did was just change this guy, and everything else, all this code is exactly the same as before. We just query, download the maths, and then we want to make the maths nice and clean, and I'll reread it in our final math file, our pad math file, which we will be using. For this, the difference in this workshop is we want to talk about the clinical data as well too. Things for things like survival analysis, we're going to need this clinical information, such as survival times, alive, dead, things like that. So the way to do this here is you do a clinical query. We have our project, our data category, data type, and kind of the format we want, and we kind of just download it into this clinical raw. And this ends up being like a bunch of lists. So we want to grab our clinical patients, and now we have our clinical patient data here. And let's just see kind of what that looks like here. Clinical patient data, right? Clinical patient, right? So just kind of our BCR patient IDs, and your tumor sample barcode, and just some a lot of different clinical information about those samples, right? So we want to take out those two. You saw those two kind of like dummy columns at the top. We're going to take those out, slice them out right here, and kind of just select the columns that we actually care about. We're going to create a survival time variable. Basically, whenever it says alive or dead, we're going to make it into a 1 or a 0. It makes it easier for our algorithms to see. So it's just a new column we're creating basically. And then we're going to kind of clean up the stages here. So like for instead of stage 1A, 1B, we're just going to have a 1. So 1, 2, 3, 4. Just kind of simplify things a little bit. And when we just kind of clean this up a little bit, create some columns, and you see here's our result. We have our tumor sample barcodes, their vital status, and you see now here the status 1 for dead, 0 for alive, and our survival time and our age and stage. So this will all be set up for our clinical data. Now it's just we just basically just wrangled it and cleaned it up a little bit. And now we want to put this in and match it with on our math file, right? So the first thing I'm going to do is look at that math file. And what I did notice was for one cancer, there's a tumor sample. I'm in a metastatic sample. So this none of the other patients have this. So I'm going to go ahead and just get rid of this because it's kind of a duplicate for that patient. So that's all I did. And then I simplified the barcodes a little bit. And then that gave me my final math file. And then I brought in my clinical clean file right here. And what you do is you just load it in, read.math. And I just put in my math file with the one thing taken out. And I think what I did before when I created the math file, so it's the same thing. Look, math, different to clean, same thing. I updated the file by taking one sample out, and then I added clinical data, clinical clean into here, and that puts it into that basically puts in the clinical data in an easy way into that pad math. And now let's look at that pad math again and see what it looks like. Hope it's there. Remember pad math, you look at the data, the listing, and that gives you all of your information, your gene signals, and all your start, stop positions, RNA mutations. And if you go down here now, clinical data, we can actually go there, and now all of this is here now. So clinical data is now stuck on the back of our other data there. So it's all together in one file that we can use. That is great. So the first thing we want to do is, every tumor is look at, mutations that they're occurring at a higher frequency or are above chance, right? So every tumor has mutations, right? Tons and tons of mutations, but most are actually just germline or they're passengers. They're just random mutations. They didn't really contribute to anything. They're just kind of there. Just by the pressures of the environment or life, they've caused them. But for whatever reason, they don't cause problems downstream, so they're just kind of hanging on for the ride, right? But on the other hand, a fewer drivers. These are actually a small number of mutations that have mutations in the right place that do disrupt things downstream, like pathways, proteins, right? And these mutations actually are the ones that promote cancer growth. So how do we statistically distinguish these drivers from our passengers? Well, that's what I was talking about. We need to look for genes that are mutated more frequently than we expect, right? So the first thing we have to do is kind of think about the background mutation rate. So all cancers have a background mutation rate. People have background mutation rate, and one of the problems you can have if you have really, really large genes, right? They're going to have more bases to mutate. So they're going to have more mutations, right? Expressed genes may have different repair rates, or chromatin states may affect mutation probability, right? So you really have to have statistical methods that kind of account for these things, right? So the first thing we're going to do is just kind of look at the significantly mutated genes. All we care about is just the frequency, how high they're done. One easy way to do this is remember the get gene summary of the pad math. And that gives you your gene summary, and we're just going to take the first head and just grab a couple of our columns here, and we're going to basically divide our, make our frequency plot, do the divide to make our frequency, right? So here's all of our genes, mutated samples, and the frequency that they're mutated in across all of the samples, right? And what are you seeing? Well, to be expected, KRAS is number 1. Well, that's good to know. And TP53 is right there, number 2, SMOD4, CD. So the top 4 genes are exactly the genes we're looking for. So we're starting off pretty good. I don't see BRAF there, but these are top 4 genes to be expected. And then we see things like TTN. Now we know TTN has a very high mutation rate because it's a very, very big gene. There could be hypermutators can latch onto it. And throw in a lot of mutations there, and a lot of times it shows up high in the list. So does mu16, and we'll kind of look at that later. Just looking at the frequency, those look good, but we need to see actually a little bit more. So this looks good for genes, but do they cluster together in specific hotspots? So now we would say if mutations cluster together in hotspots, they're probably more likely to be driver genes, right, than not. So we're going to use this OncoDrive function from math tools. We're going to just look at minimum mutations considered. 5 has to have at least 5 mutations for in a patient for it to be pulled. or 5 mutations overall to be pulled, right? If you don't have, you have to have over 5 mutations over the whole, out of all the samples to be pulled, and then you're looking at just the math, and you're looking at the protein basically changes, and we're getting that file. We just kind of look at the significant genes. We're taking the FDR under 1 from 0 to 1, I guess, because you want an FDR below 0.1. And what do we get? Well, we basically get 2 genes here that are significant. One has a pretty much a low number of genes, but they must be put together in a hotspot really well. It's the smallest amount. But they're still significant. But KRAS jumps out as the one you really jump off. And once we have our significant genes, so all we did is we got significant genes, we filtered for FDR, and that's it, really. And then we put it in here. The plot, we can plot this significant genes, and the result here for OncoDrive with FDR cutoff of 0.1, and we see our 2 genes are up here. So what's happening here is the X plot is a fraction of mutations that are found in clusters. So the higher this fraction, the more are found. So the more found in the clusters. So that is worse, right? And then FDR, the higher, the more significant, and size is the number of mutation. And so our red ones are actually significantly statistically significant drivers, and as we would expect, KRAS is the highest one, which makes sense. But the other ones didn't work out too well. So that's great. OncoDrive basically identifies drivers based on that positional clustering, right? But that's not really the gold standard to do this. We really need to look at something a little more robust to really find out our answer here. And one of the gold standards in the field to do this, there's several of them, but this DNDSCV works really well. It's being used in a lot of publications lately. And basically it works by observing kind of the ratio of non-synonymous to synonymous mutation rates, but it also accounts for the trinucleotide content, right? So you have the 5' and 3' flanking base pairs, as well as the mutation, the sequence composition, and also the covariates. So this is just a very good, robust method to really tell you, are there more functional mutations that are expected by chance, right? So what we can do, we got to make our object to kind of fit what it wants to look like. All it needs is this information, and we have it here on the left, and it's expecting the names on the right. So we're just grabbing our map data file, we're selecting these columns, and we're just renaming them. Simple as that. And another thing we're doing is the chromosome. If you look in the map data file, I remember if you look at the data, see how it lists chromosome CRH1? Well, this program needs it to be just 1, 2, 3, and so forth, right? So what we're doing is basically we're taking out the chromosome, we're leaving the number, right? And then once we have that, we have our file to put in, and we have our our sample, our scaffold of 38. And then we just run our simple function here, get our readout, and then basically if you look at this cell CV column, that's where you get your result. And basically we're just filtering by a global Q value, which is kind of the FDR. Just think of that as if you're below 0.05, your multiple testing is corrected for it, so it's kind of like an FDR. And then we've kind of selected out some of our columns here and just taken the first 10. And you can see they're all have really low Q values and high some of these ratios. And what comes off, once again, TP23, KRAS, CDKN2A, SPON4. So we're once again seeing the genes we want. So the fact that we're seeing them in several different places makes me feel a lot better about them as far as they're frequently mutated. At least KRAS is found up there, and then OncoDrive plot, and then now we're seeing them statistically found here. So these are definitely the driver genes. We feel pretty good about it. Here's some different methods and approaches, but this is really one of the current standards here to get this done, and it really makes a lot of sense to define these mutations that are more expected by chance. So now that we have that, let's talk about the co-occurrence and mutual exclusivity analysis for this data set. We've done this before. We're just we introduced this last workshop, and we're just taking a little bit farther. We're going to use map tools at the beginning. And if you remember, the co-occurrences, mutations in the same pathway may occur, or mutual exclusivity is mutations in the same pathway may be redundant. So if you have one, you might not need the others because it's already been accomplished what cancer wanted. What cancer, like it's a person. So let's analyze this. So let's do somatic interactions function from the math tools. our package. We just put our pad math. We grab our top 25 most significantly mutated genes, or the top frequency mutated genes, and we kind of look at them all here. If we see a small dot, we are below 0.05. A large dot is 0.01. So let's jump right into it and look at KRAS, right? KRAS, TP53, and CDK2A are all co-occurring. So they like to co-occur. TP53, KRAS. But interestingly, if you have all three of these, if you have KRAS, mod 4 is a little bit, it's in there, but it's not significant, right? So that's interesting. And if we kind of find some more of the genes that are interesting to us, really those are the main ones. These four here. And you can see like SMOD4 and CICNB are co-occur, GNAS, TP53 do. KRAS is a little bit, it's interesting that GNAS is mutually exclusive to both these guys. I know it's also showed up as a driver. So it could be that GNAS kind of drives that RAS pathway if you don't have KRAS TP53. So these are kind of things you can think about and look at to see if things co-occur or they're exclusive. But I think the main thing is our main ones do co-occur. And that's definitely interesting. So let's just break this down a little bit. So I just grabbed this to a little bit smaller. I basically just made this a little bit smaller to the top 20 genes, and then I put this into interaction. I made an interaction object of this, and I basically just filtered for p-value, and should have filtered for p-adjusted value, but and I just kind of looked, kind of give you a summary of what you're seeing here. So like TP53 KRAS they co-occur with a really low p-value, for example. And you can kind of look through it, and it gives you an idea to see the top 15 different interactions. So that looks pretty cool. I think that's a really good thing to do. But like, the problem here is, so somatic interactions, it runs a pairwise Fisher exact test across all of your top mutated genes, right? However, it has two key limitations. It doesn't account for the background mutation rate, which were things like hypermutated samples, other things can drive, spurious co-occurrences, and then also treats each pair independently without properly handling the massive, multiple testing burden. So, as you see here, it does correct for p-values, but it's kind of after the fact. It doesn't really model it while it's running it. And where Discover actually does. Discover is really a better package or approach for a more robust co-occurrence analysis. So like multiple testing correction, if you have a bunch of mutations, right? Well, we're already saying like the standard is to say like p-value 0.05, right? So you're already saying that if you do something 100 times, five times it's going to be false. So that's one problem with genomics. There's always going to be these false positives, right? And the more samples, the more times you test things, the more likely you're going to have a false positive, right? If you have 1 million samples, 5% of that's going to be a lot, right? 500,000 or whatever. And it's really going to matter. So, or 50, whatever, I don't know. So it's really going to matter. And you guys know that if you do ANOVA or you do multiple t-tests of like 5 samples, you have to correct using, Hushberg or Bonaferi, things like that. So this is what we're doing, is kind of baked in here. This program has an ability to handle that better, where we can report our FDR or our p-adjusted value, same thing. And you can have a little bit more slack on the FDR. 0.05 is still the standard, but you can go to 0.1 still sometimes. But so one way it is handling all of these issues is Discover is modeling the per sample mutation rate as a covariate itself, okay? And so two genes that are both frequently mutated together will appear that they're co-occurring in a Fisher sample test if they don't have this covariate, this model. So that's how it can really help account for it. There's other things too where it accounts for some of these things under the hood, and just a little bit more robust thing to use. So let's go ahead and try Discover and see what we get here. So what we're still going to do is build our mutation matrix. We need a count matrix this time. So we need a matrix that has like zeros and ones. This is actually a really good function, math tools mute count matrix. And you run that into math, and let's see what this looks like, because this is actually very useful for other things. If you want to do clustering or phylogenetics, So this is perfect. So we have all our samples here as columns, all our genes as rows, and just a 1 if the mutation is there, a 0 if it's not. This is a really, really useful matrix to be able to pull out to do a lot of different downstream analysis. So that's really cool. So we have our mute matrix, right? So I've already like fixed it here, but we want to make sure if we see that that there ends up being a true or false to that matrix, what can happen, you could take that matrix and basically just say if it's over 0, put a 1, which means it's going to put a 1 for the trues, right? And everything else is going to have a 0. But it looks like it already kind of comes that way, so we're okay. But just to kind of know that if you have true, false, you need to change for 0s and 1s. The first thing we want to do is estimate the background probability. So we take our matrix and what is going on with that bin matrix? All right, so this is the same thing. So basically, this is kind of redundant because it came out, but sometimes some maps don't have that information. That's why you have to change it. Basically have our mute matrix 1s and 0s, minute numeric, and so you get PM, that's our background probabilities, that's our probabilities, and now we run our tests for mutual exclusivity and co-occurrence, and to do this we go get mute X, we use our matrix, and we put our probabilities we put in there, and our lower dot tail equals true for exclusivity and false for co-occurrence. And then we're just getting our gene names here and just kind of making sure that all these have the right gene names. And we will then extract significant pairs. So we're basically just looking and basically pivoting to longer here. And we're just basically grabbing out the, filtering for p-values under 0.05 and adjusting the p-values as well. So once we have all those, we have our two results here. So process discover is a function that we made here. Just all this is doing is filtering out p-values under 0.05. We have our results. We're binding them together to this all results. And then we're making a new column called p-adjusted. And here we're doing the Benjamin-Hochberg method to correct for our p-values here. And they're already set up better for this to work. And then now we're just displaying all our results. And then we're just applying our top results here. And you'll see you have a gene 1, gene 2, you'll have a p-value, adjusted p-value, and if they're a co-occurrence or exclusivity. We don't see our top genes up here because I didn't sort for that, but because we have all of them, right? So the thing to do now would be to basically filter out the columns for our top 20, 25 genes, or even the top 10 that we like, and be able to check here and match to see if they're adjusted well. But this will give us a much more robust answer to our question than the one above. But I suspect if you look into it more, you will see the same result, or very similar. All right, so now we've kind of looked at our exclusivity and our co-occurrence. Now we want to talk about those hypermutators. We mentioned TTN and some issues with these hypermutators and how they can really just mess things up. So what does this even mean? So most pancreatic cancers have a very modest mutation burden, but there's a small subset that are hypermutated. They have 10 to 100 times more mutations. They're just gone crazy. There's a lot of things that can cause these kind of things, from APOBAC to POL to cell cycle issues. But why does this matter in our cancer? Well, what's interesting is normally you would want less mutations, right? But in this instance, we kind of want more. So if we have ultra high and ultra high mutations, so our TMB, per megabyte is like over 10, over 20, which is a very low population, but this actually gives us a better chance for our immunotherapy response. So since this cancer is so hard to treat, having more mutations is actually giving us a better chance to treat it. So it's very kind of counterintuitive, right? But this is obviously for our current methods, right? It doesn't mean you want more mutations, but our current methods are the ones you got to use, right? So let's calculate our TMB and see if we have a hypermutated sample, right? So remember the exome size is about 38 megabytes for our TCGA exome sequencing. And they're basically going to get our counts, and we want only our non-synonymous mutations for this. So we want to do our MAD data, and we want to count all of our samples that just have mutation count, right? Because we want just non-synonymous, right? If you want all of them, we could do a row bind of PADMAF, the PADMAN silence in there, right? And we could put those mutation counts. So you want everything you can do there. We're going to go with just the non-mutagenous, and we want to merge all samples in a way that we can catch ones with 0 mutations, because we do not want those, right? Or we want to be able to account for them. So we merge all samples, we left join all our mutation counts back to our data, and we have a mutation count, right, column, and we replaced NAs with 0. So we don't want NAs, we want all those with 0. NAs, we want everything without a mutation count to be 0. And then we calculate, we have a new column for TMB, and we just do our mutation count divided by 38. That gives us our TMB, right? And then we have another category that's basically saying, depending on your TMB, what category will we put you in, where if you have over 20, it'll be an ultra high, for example. So we're basically just wrangling this together. We basically just did our mutation counts, divided by 38 to get our TMB, and then kind of just wrangled this together, and we wanted to see basically the TMB of each sample. That's it. So now we have a TMB summary that gives us our TMB. Now we have our TMB data. If you want to see what that looks like, we can go check that out. Our TMB data, or sample barcode, just our mutation counts, our TMB and the category. Real simple, right? Just data wrangling here. And then we basically just we make a new column for our percentage to see what the percentage of that is, and then we output it. And we're basically seeing, we had a pretty low cutoff for TMB, I guess I could have put a little higher for, but really it's really not bad, because if you think about it, the low ones are under 5, so most samples have under 5 mutations. It's crazy, right? This guy is a lot, over 20 for sure. So it's one guy, which have one really high hypermutator in this data set. So that's really good. So that's how you would check for hypermutators. We only have one, but depending on your cancer type, you may have others. This is the process you would go about, right? What are other things we care about with hypermutators? Well, one thing that can cause hypermutators is microsatellite instability. And this is something we would always like to check with our samples to see if we have the amount of mismatch repair we have, because this can happen in a cell cycle where errors can accumulate during the DNA replication process. It can create microsatellites and instability. This can really cause your mutation rate to go up a lot. So one thing you kind of want to check on if you have a lot of hypermutators, or if you want to just check on in general for MMR, is you will want to look for these MSI at different levels. And basically, one way to look for them is what genes are associated with this, right? So we know we can look for MSI high. It's kind of what we want to look at for the most efficient, right? And we see here there's 4 genes here that are MLH1, 2, MSH6, and PMS2. They're very associated with MS, with microsatellite instability. So one thing we can do is go check for these genes. Now, if we have these genes, this does not mean that we have MSI. It means that it's probable, and we need to go a little bit farther, check for other things like mutation signatures, things like that. Like you expect to see 6 to 15, SPF 6, SPF 15 if you have MMR. So there's different levels of redundancy and things you can look at to make sure this is actually happening. But the first thing we're going to do is just merely check for these genes. Are these genes in our data set, and how many are there? So we're looking at our data file once again. We're going to filter out for the gene signals of all these guys, and then we're going to select these columns for them and then see what's up with them. And we're going to check our uniques, and we will see that there's one sample, one has these genes. And let's just check out which one it is, and it's the 7651 sample, and it has, 1, 2, 3, 4 of these genes, 4 of the 5 genes it has. And here's just some kind of things it has to do, different proteins and variants. So we basically found our hypermutator here, but is it actually MSI? I don't know. So let's actually look for mutation signatures in the data now too, to see if we see those. So to do that, we're going to use SIGMINER, and we'll talk a little more about mutation signatures and SIGMINER next workshop, actually, in our 3rd workshop. But I just want to introduce this here just very briefly, just to be able to check here to see if this is correct. So you don't have to worry too much about how I calculate this stuff. You can check it out if you want, but we'll go over this again next workshop. Basically, I'm just looking for signatures and see what's up. So first I need to do is signature tally, sig tally, pad math, reference genome, mode SBS, right? That gives us our tally. And then what you do is you call these lists of all matrices, SBS 96, and that is our input matrix. Then we do something called sig fit, with that input matrix, we're looking for all signatures with our latest build, and then we're just going to sum those exposures across all samples. So we want the signatures across all samples to kind of squish together into one, and then we can also look at the more active signals. You could have just put fit here, but if I want to choose specific things to look at, I can be like, oh, I want to be strict and keep only signatures with over 1% contribution. They have to have mutations that have over 1% contribution to the whole data set, right? Because I want to show they're active, not just show everything, right? And if we do that, we can run our show sig fit, which is getting our fit from up here. And then, that's why I calculated total exposure to figure that out. So we basically just put our fit from sig fit up here, and then we do our active signatures that we want to look at, and we just plot them down here, and here's what we get. And we see here, one's kind of an anti-aging to be expected. 10 A and B are hypermutators for pole mutations, so that's interesting. These are kind of fluff probably, but what jumps out to me is 6 and 15. 6 and 15 are the MMR signatures, so that's good. So the fact that we see that hypermutator, it has the right genes, and we see these signatures in the data set, I would say that we can say that probably has MSI, right? So let's look at actually with the TMB of that sample. So there's only one sample, so let's just pick it out. And if they're mutated, we have, 335 mean TMB, right, for that mutation, right? So there's a lot of mutations happening in that mutation, and the mean medium is the same, right, because there's only one sample. So we're only expecting about 1 in 7,300. So that's a lot, right? So let's talk a little bit more. Let's look a bit at our poles, because poles could have caused this too, and then we'll see if that's in our data set as well too. So pol and POL-D1 encode DNA polymerases with proofreading activities. You can imagine if DNA polymerases with proofreading activity have mutations, that could really mess things up really bad and lead to a lot of different mutations. So let's check the same thing. Let's look for these two mutations in the data set, and lo and behold, we see pol in the same data set. We also see it in one more here, A919, so that's interesting. So there's two different ones that have them in there, but we see the one we really care about is happening there. It'd be interesting to know the TMB of that. I should have looked at that. We could too, right? A919, 3A1. Here it is, 86. So that one has 86 total, even though it has pol, which is still a lot, 86 mutations, considering what is the average? Well, the mean was like 1 or something, right? So a lot of these have 1, 20, so the mean is more like 20 or something. You're thinking of TMB. So the mean somewhere around 20 or something mutations. This one has 86, so it has 4 times as many. So it kind of makes sense that that pol mutation could be causing this guy to have hypermutation. But this guy's the main guy, right? That's really causing the problems. Let's kind of just visualize this hypermutators. And there's not many of them, right? There's only one. So you can really see it really drops off fast. It's kind of a trivial plot, but it's just kind of pushing in your face that there's only that one, right? So but I want to give you the code here. So the idea is here, I'm using this for PAD, but what if you want to look at a different cancer or your own data set? I want you to have this pipeline to be able to pull out and visualize these things, even though this one's kind of trivial. Make sense? So now let's find out who the hypermutator samples were. We already knew pretty much, right? So let's just look we're basically going to grab the 10 most mutated samples and, check that out and name them. And here they are. And, there's the 10 most. And this guy is just obviously the ultra high. These are kind of not that bad, but worth keeping an eye on for downstream analysis for how many mutations it actually has. And there's our other pol one right there. So, all right, so just kind of throwing this in the context of what the TMB should be. We're around 2, right? 300 is the high. So it kind of makes sense for us to be around here. If you look at the whole TCJ set, we're kind of on the low side, and you see my pad came up right with a real one. So there's mine, there's it, and we're hitting around 20 or so mutations, our TMB. So ours is a little bit lower, but it's getting pretty close to what it should be, right? All right. So now that we've looked at the hypermutators, let's move on to one more thing we can do. What can we do with clustering? Can we cluster based on mutation profiles? Why is this interesting? Why we want to do this? Well, patients with similar mutational profiles may also share underlying biology, right? And that may cause them to respond to similar treatments, have similar prognosis. And so we want to see if these things cluster together based on their mutations, right? And obviously other things too, but we're the mutations for now. So let's just check that out. So we need to have that binary matrix, right? So first we want to do is we want to get frequently mutated genes present in least, we want mutations that are present in at least 3% of the samples, right? If it's really highly frequently mutated and that hypermutated one, we don't care about it. So we want to kind of protect for that. So we're basically grabbing our samples and we're setting a ceiling where it has to be in at least 3% of the samples, really a floor, really. But anyway, we're basically just filtering out the data, grabbing the symbols, grabbing the barcodes, distinct barcodes, and then we're basically just saying we want samples that have greater than the minimum amount, and then we're just throwing those, and we have 42. If we do that, we have 42 frequently mutated genes that are found in at least 3% of the samples. So that's going to be what we're going to use for our actual clustering. And then from that, we are going to basically make that binary matrix. So we have our data right here, right? We want to filter out the gene symbols that are in frequent genes. So our 42 genes, we want to filter those 42 genes out. We want to take the distinct, unique barcode for each one of those, and we want to change the word mutated to 1, right? So we can have 0 and 1. So 0 for wild type, mutated equals 1. So values from equals mutated, otherwise 0. And we basically just pivot to wider because now we want to pivot to wider for the tidy stuff and convert to our mutation matrix, actually our final mutation matrix that we will be using here. And that really what we're doing is just filtering. What you could have done is done that earlier when we could have made that mutation matrix. Just like before, we pulled out with that mutation count. Remember that function? We could have done that, and we could have just filtered for the 42 genes. So this is just another way of going about it. But all I'm doing is I found I found the 42 most frequently mutated genes that are found in at least 3% of the samples. So I basically just found these genes, mutated in at least 3% of the samples, and then I just took those out of the data and then basically made mutated 1, wild type 0. That's it. And then now we have our mutation matrix that I've showed you before. That's actually mute matrix, but it's the same thing. Mutation matrix here. And now we have here, as you see here, this guy has 159 columns. It has wow. So it has 9,000 genes, right? And this guy has 142 genes. So they're kind of flipped here. But here are our samples. Here are 42 genes, and they're ones and zeros. So that's perfect for clustering. Perfect for phylogenetics. That's kind of a great matrix you can do. Another thing you can do is do like if the mutation is homozygous, put a 2. So that's another level of complexity you could add if you want. Another thing we often do, just to tell you, is you can also cluster by variant allele frequency, which can also be useful too for more refinement. But anyway, now we have our 42 genes. There's the 42 most frequently mutated genes that are found in at least 3% of the samples. And now we have a mutation matrix, so we're ready to do our clustering. And clustering, we kind of go over this past in the last semester and stuff, but just really quickly, Jacquard and binary are very, very similar. And basically, those are the best to use for very sparse data, and especially like binary data. Or things like Euclidean is very good for continuous data. It's good for actually RNA data, things like that. And Manhattan is really good for, it's very good at outliers, things like that. And Manhattan's actually really good for copy number. That's kind of the way that I work with them. But just to show you here, we're going to show you the different ways you could do this. When you're doing clustering or trees, you have to calculate your distance first, right? So we're going to calculate our distance using our mutation matrix and binary for Jacquard, and we get our Jacquard distance here. And these are other ways we could have gone with it. And then we do our H-clust. To do our hierarchical clustering, we have H-clust. We bring our Jacquard distance, and we're using the Ward D2 method, which is the most common method to be used, to be honest with you. You could have gone a different way and used complete here. And you can check out the difference yourself if you would like downstream. like didn't complete, for example. I think for us, the best thing to do is just to go with our award. It's the most widely used. So this Jacquard and Ward are the best for our data set, I believe. So that gives us our H-class object, and then we want to visualize that in a dendrogram. So we say as.dendrogram, HC Ward, den Ward, and now we want to plot that. And what I've basically done, I am choosing to have to cut the tree, right? It's the tree, hierarchically clustered tree. Well, to make the clusters, we have to cut the tree. And I'm basically just going to start, I'm just going to guess that it's 3. There's no reason for me to choose 3, I'm just choosing 3. If I looked at this data and just glanced at it, I would have chosen 3 anyway. And you could also cut the tree by height. So let's say we cut the tree by 2.4, it would go across, and wherever it cut, it would grab a cluster, right? If we cut the tree like at 2 or 2.1, we'd have 1, 2, 3, 4. So we got 1, 2, 3, 4. That also makes sense. I could also see a 1, 2, 3 cut maybe there, maybe there. Yeah, it's really hard to cut 5. 1, 2, 3, 4, 5, 6, 7, or 8. I could find. So like 3, 4, 7, 8. Just looking at it, but 3 is pretty solid, so that's why I chose 3. And then I'm basically just coloring them and plotting here. So plot. I basically just color branches function, den Ward, k equals the number. That just colors the branches with the in-house colors. I didn't choose the colors, and then I just plotted it and we're colored, and here we go. We have our 3 different main clusters that we saw here, which I think is pretty interesting here. But how do we, if we really wanted to know how many clusters, how would we do this? Well, this is all covered last semester. Just to remember, you can do the silhouette, which measures how well each sample fits into its cluster. And I'm going to use this SViz MBClust to basically do each one of them because it makes it really easy. You can just put in the Jacquard distance and your H-cut object. And once again, your Jacquard distance here and your method of silhouette. And basically you just change the method. See method silhouette, method WSS. This is just a label, gap stat. So you can try a lot of different things here, right? For elbow, it kind of measures the within cluster compactness. Gap statistics compares your clustering to a null reference. Okay, these are all just very standard ways of looking at things. And then finally, we do this consensus where it basically runs 30 different things on it. You just throw in the mutation matrix, and we're going to use Jacquard distance, Ward 2, or D2, and it's basically gives us a million different things. And we're going to skip this part here. This is kind of in-house things that MBClust does, but I think we'll just kind of just skip to the end of that, and basically it went and looked like 30 different things, and it found a certain amount of consistent ones, and basically you see here 3 different methods said 2, 7 said 3, 5 said 4, 5 said 8. So there's a lot of discrepancy among the different methods. This is kind of a hard choice to choose number of clusters. But looking at kind of the in-house MBClust, it's basically by majority rule, it's going to be 3. But let's kind of visualize this a little bit better. So I'm pulling out that stuff I'm basically pulling out the different results, and I am plotting them here below for us. Because I pretty much already see the PVS, P-sil. These are already made, basically, these objects, right? So all I had to do is come down here and basically make our NB plot. So I take out basically the best one is 3, put it in here, P and B, and then I put all of them here together, and we see all of them great here. So I want you to look at the bottom right one first, I think. So that's the one that looked at all of them, the most statistically robust, and it shows 3, but also 4 and 8 were high. If you look at our other methods, they don't work that well. So, silhouette is saying 4. Elbow is saying, I guess, 8. I don't know. I guess you could say 4. It's hard to know, like when the elbow happens, it happened at 8, 4. So it could be 4 or 8, 4. So 4, 4, 8. And gap stat is saying 8. So we're seeing 4 and 8, right? Which is kind of what we're seeing here. But it's saying 3. So it's interesting, right? But this is the best method. So we're going to go with 3. And basically want to visualize this now. And we're basically cutting the dendrogram. If you look here, I got my optimal. That's just going to be 3. I'm just looking in that data thing, and then I'm cutting the tree at K to get my clusters. And it's really the same thing I did up here, right? I guess H class chooses that you don't cut it up there. But yeah, so I'm basically just cutting the tree to get my 3 clusters. It's basically just giving me my sample IDs in those 3 clusters. And then we're going to create an annotation for the heat map because we want to add the TMB to it as well. So we just want to add if it's low or high TMB, and then we add in our cluster information to our annotation. So basically we have an annotation that we have our barcodes, and we've joined our barcodes, and we're putting our TMBs, and then we're adding our cluster information like 1, 2, 3. Then we're adding our colors, and we're making our feet map. Before I show it to you, I'm basically just putting my mutation matrix, all the 1s and 0s, and I'm saying I just want to cluster columns only. We're doing it exactly the same, binary and word 2, and we want to keep our same clusters as before, and we're just coloring, simple as that, and we are basically clustering for both of them, but we're keeping our clusters from the hierarchy clustering on top. So they're going to match exactly, because that's all hierarchy clustering is the top of this deal. So we have our 3 clusters, and then we have our other mutations that are clustered on the side here. And then you see our 3 clusters, and then we see that one little hypermutator there. So you can see here all these mutations along the whole thing. So it's pretty obvious that's the hypermutator. So what jumps out here? What separates these clusters? So this is always a good thing. One of my favorite things to do is a clustered heat map, and take a look at it. So what is happening here? So blue doesn't have TP3 or KRAS very much, right? Or SMOD4. It doesn't have anything. So it has just a bunch of junk here with a couple of mutations. So that's kind of our cluster there. It'd be interesting to see making a tree and see where this fell out on it. Our green cluster, we have almost all of them KRAS, almost all of them TP3 as well. But we do have SMOD4. So SMOD4 is not seen much other places. But it is seen a lot here. So that's what's separating this cluster out itself. And what's going on with the red cluster? Well, once again, we have mostly KRAS and T53. And that's what separated it out, I think. What separated this thing out, I think, the most is not having, these have nothing, right? Or pretty much nothing. So that's why they're separated. What's the difference between this and this? So what's different? Why green separated out was because of this SMOD4. So that's why you got all of this. And then all that's left is the T3 KRAS, right? So what's different about these 3 clusters? Because I see 3, I just feel like I see like one. So you get one there, right? So there's a cluster here, right, that has this TTN, mu6. So this is kind of the hypermutator cluster, I would say, right here. And then obviously CDK2A, all of this guy, it has KRAS, TP3, and CDK2A. That's that cluster, right? So this guy has SMOD4, this guy has CDK2A, this guy is the hypermutator, and this guy just has KRAS, TP3, but not much below, right? So that's how you can really see, really break down what is going along in there. And if you want to look at the clusters kind of in general, they have kind of similar medians. The mean's higher because that one hypermutator, and which genes are in each cluster. We kind of talked about that. And cluster 1, we have the KRAS, TP3, and CDK2A. Cluster 2 is the one with the hypermutator. Cluster 3 is the one that has SMOD4, right? And that's kind of what we're really seeing that's separating out those clusters. I think it's really cool, really good illustration of what you can do with clustering. So let's move on. So now a new question is, we've looked at all these mutations, we've seen the important ones, but do these mutations actually predict patient outcomes? How does this look at their survival? If we look at their survival data, are we going to see they have an effect? Okay, so let's do that. So the first thing I want to do is merge that mutation data with the clinical data. And I'm just kind of cleaning up that data where I want to just look at these key genes. So I'm basically making a new data frame called mutation status. And I'm basically it's very simple. I'm bringing in the data. I'm filtering by just genes. And I want to know if they're mutated or wild type. And then the next step is going to be to add the TMB status, and then add the mutation status, TMB label, and I finally will have my actual survival data. And I'll show you. So it's just wrangling. It's just basically wrangling and joining a bunch of sets we already have, and what you'll get. And in the end, see, all you do is take the clinical clean, and then you want to interjoin that mutation status, and then basically filter for our survival times. That gives us our survival data. Clinical clean, just to remind you of what those things look like. Clinical clean is where we have our important data, right? Our zeros and ones, our vital status, things like that, days 2. So that's really important data, right? And we'll bring that in with our mutation status. Mutation status. So we're bringing this in because we want to know, this has been, there's not as much in this mutation status because I moved it down, but you basically want, for each gene, you want to show, I recreated this matrix later for another thing. That's why I stopped telling everything. But basically, you have your barcode and you have each gene. And for each gene, you'll have all of them. And you'll have, if they're wild type and mutated. And then finally, we can, work on the survival times, change that to the right. And so that you do all that together, you'll have our survival data. Hopefully, survival data. To show you what that looks like. Yeah, here we go. And that kind of just gives you all your survival data. Remember, you have your ones and zeros in there. Survival time, ones and zeros, if things are mutated or not. Simple as that. And one reason you're seeing it like that is because we pulled it from an earlier deal there. So anyway, so now we have our data here. Let's actually fit this data. So TPT3, errors, and SMOD4. Okay, that makes sense. So like I said, that the reason it doesn't have all those columns in there is because I recreated the data set later to do a survival analysis later. So for sake of time, it's going to have a column for each gene, obviously. How could it not? So, so basically, you're seeing there's this, and there's 2 more columns for SMOD4 KRAS, right? So just realize that is all there on the one you put in. Code's all right, no issue at all. So we're basically putting out survival data, and we're saying for TP53, we want to look at the survival time and the status. That's all I really care about. Because we just care about if they're alive and the time, right? So if you look at TP3, then we're going to plot this using GG survival. So I basically have this survival object I create, and then I have the survive fit. And I get my final fit, and basically I put in my survival data, my fit, and then just some different parameters for the plot, and we see we plotted here, and we see there's 0.01, right? 0.001. We have a good p-value. It's mutated. The survival seems to be quite worse, right? And let's do the same thing with KRAS. It's even less. KRAS, if you have KRAS, it is even lower. And so there's even a stronger chance of the survival is definitely even lower if you have KRAS, right? Let's look at SMOD4. Same thing with FOS4. FOS4 is not significant. In some cases, it's actually even higher, right? So SMOD4 does not, if you have SMOD4 alone, that does not say that you have worse survival. Where these say if you have KRAS or TPT3 alone, we're just looking at this alone in a vacuum, that your survival is worse. So that's the vacuum, right? Well, how can we look at this in 2 different ways? One, we want the odds ratio. What's odds ratio is, odds ratio of 1 means it's not good or bad. Anything higher than 1's bad, anything lower than 1 is good. And we also want to look at this across several different factors at the same time. So we're basically grabbing our survival data and just formatting it and taking out this unknown column. And then we're doing our survival object again, but this time we're doing Cox pH for our Cox proportional hazardous regression. This time we're putting everything in, because we want to know if you look at everything together, is it significant, right? Not just a vacuum. So we put in the survival GG4, put it in there, And we're looking at all these things together. And here's our result, and what do we see? I only see something significant, really, in the KRAS. So if you have KRAS, it is worse. Also see age and stage. They're kind of nominal, right? They're almost significant. So worth looking at, but we'll have to look a little further. So this kind of just gives you a summary of that stuff, but the best way to do this is a forest plot. And you can just do your Cox model and set these both to true. And then I am cleaning the term names, basically. I just changing things like stage symbol. I'm just changing up some of the names, making them cleaner, and then I am adding the significance. But I'm basically just plotting those things into the forest plot, and then I just drop my forest plot, and let's just look at what we got here. So once again, you see if they're near 1, they're not better or worse. So even if they are significant, like age, that it doesn't really do anything anyway. But stage showed close to significance, right? So stage 2 shows worse, and it's almost significant. So I would even say that looks good to me. TP53 and wild type so it's saying wild type is better. So that means if you have non-wild type, it's worse, right? So it's basically saying it's worse if you have a mutation. KRAS is the only one that's significantly worse. No, even in the term of looking at everything at once, there's with all the covariates, it's still significant. TP3 is still almost significant. What was the T3 wild type? 0.13. So it didn't have a really good p-value, but it shows a good odds ratio. So something to think about. But we know really stage and KRAS is really showing a pretty solid effect, right? So now we kind of know what's going on with survival. Let's take another step further and look at the pathway analysis. We're going to look at how individual genes, tell only their part of the story, right? That vacuum, right? Now that we've extended the mutation survival vacuum out, let's consider the pathways, right? And look at how the network of genes work together to disrupt this, biological processes in this cancer type. So there's obvious benefits of finding these pathways. You guys well know. Most importantly, just to understand what's going on, to develop therapies, right? And sometimes you have a lot of genes, but you don't really know what pathways they affect, or there can be a lot of crossover. It can be kind of complicated. So you want to really look in there and see what is actually happening. One quick overview way to do this is using the math tool stuff, like always. You have the oncogenics pathways function. So it just jumps out and gives you a lot of things really fast. You see things like TGF beta and the RAS pathway, things we expect to see. You see the sample of fractions. See, I really just see TP3 pathway is really disrupted, and the RAS pathway is really disrupted. So it makes sense, right? Because we have TP3 and KRAS. And if you want to look at just the RAS pathway, you can do plot oncogenic pathways and just choose one of your pathways. And basically it's saying that almost all of our samples have KRAS, but not many have the other ones. To be honest, it's kind of interesting. That's the hypermutator right there. A lot of them have both, and there's some, mutual exclusivity for some of these guys. It's kind of interesting, like EBBR2. There's one patient that has it, and it's exclusive, mutually exclusive from KRAS. Anyway, it's just kind of a really quick way to get a look at it. But we want to go a little further into this, do some gene set enrichment analysis, try some different packages that are commonly used, right? So let's get started on that. So how can we do that? Let's grab all our mutated genes first. So I'm just looking at all our genes. By the gene symbol, I want unique. I don't want duplicates, right? So we have all our genes. And then I basically want to change all our samples to a different name, right? So we have Hugo symbols, but we need interest IDs for what I'm going to do here with the Go enrichment analysis. So I'm basically just changing, I'm flipping the IDs. No, not that big of a deal. So I just have my mutated genes, and then I flip the different IDs, and then I'm going to put them into. And I'm looking at all the genes. You could look at you could just, I'm just giving you an example of how to do this, but you can also choose your most highly mutated genes, different sets, right? But the more genes you have, the better these analysis works. These are over-representation analysis. It's not like RNA-seq where you're doing different groups and seeing which one is different, right? Do the genes have statistically different pathways than this group, right? We're not doing that. Now, what we're saying is, do we have genes that are mutated in pathways, specific pathways, more than chance? So they have an overrepresentation in that pathway. That's what we're trying to do. And we're not just matching and saying, hey, do genes match pathways? They have to be in those pathways more than background to be expected, right? So now we have those genes converted. So the more genes you can do, the better. But remember, you can choose, to choose less if you want. So here we have all of our mutated genes, and we just put them in this enrich go, and we want to choose the biological processes. Now we could also do the cell components, right, or the molecular function. There's 3 different ones you could do here for the enrichment, for the go enrichment, right? So we're going to choose BP to have 1. We're doing adjusted p-value by BH, and then we'll go ahead and plot it. And we see here the different gene ratios, the counts, the adjusted value, and some of the the biological processes that are affected by this. We could also do this pretty much the exact same thing. We just do enrich KEG instead of enrich go, and we get our KEG pathway enrichment. We can also do enrich pathway. This is all cluster profiler. We're just basically just changing the top, and we can put in different genes if you want, and that's giving us all that. And now I'm just going to kind of look at some of these guys and just see. Now I'm going to see just going to tell you what genes are associated with what pathways, right? So, for instance, we have 108 samples, which are 64% of all the samples that are affected by the RTK RAS pathway. I can tell you they're mostly RAS, but we also see DNA damage response because TP53 probably, and TGF-beta is also really high, and cell cycle is also pretty high. So these are all ones that we would expect to look at, expect to see up there on top when we see those different pathways. So these are ones we kind of expect to see. Then we go through our different specific matches, and especially like hallmark and stuff, and see if we see RAS, things like that. So that's one way we could have done it. I'm just going to show you another way. So I showed you the math tools way. I showed you the cluster profiler way. Now I'm going to show you the FGC way, the fast gene set enrichment analysis. So I'm just kind of showing you different ways of doing things, because oftentimes one's better than another, or different results. You want to do it over and over and see across data sets if you're seeing the same thing. So how do we do this overrepresentation analysis here with FGC? So here we have to make our sets first. So if we want to do our hallmark sets, what we do is we use MSig DBR to pull all of our sets to match, right? So we do species Homo sapien, category H for hallmark, and we just pull all of that for hallmark sets. Same way here for the reactome, we use category C2, CP reactome, and you basically just change these different values, the categories and subcategories to get any type of thing you want. So it's much more flexible. You can just pull specific pathways to match, and then you basically just make our genes into a vector, and we actually rank them now. So here's something different. We want to rank these. So we're telling them these genes are more important, right? For whatever reason, for frequency, functional impact, we're doing it by frequency here. So that'll change a little bit. Before we're throwing things in unranked. That's why I said you could subset for the best. But this is the best way to do it. Put every gene in, but rank them. And then we run our deal. We throw in our pathway, whatever pathway we want to choose. I'm going to choose reactome and our ranks and just run through it, visualize, and here's what we got. We see all our different pathways and FGCA results. We can check all our different pathways and all our repeat adjusted values, and we get to see the genes that are associated with each one of those. So this is another way of looking at it. If you want to visualize one of these, if you want to visualize any of these, you can use Enricher. It's an easy way to do it. You grab your data set, your hallmark DF or hallmark sets, whatever, use Enricher, use that vector of gene, and we're basically just changing the gene signal here, and then we're doing our adjusted p-value, but it basically just enriches another way to be able to plot these guys. I just didn't plot these guys. You can also plot out of FGCA pretty easily too. It's just a different way. So there's lots of ways to do this, right? You can use algorithm. You can use the Go process. You can use cluster profiler for like Go, KEGG, Hallmark. You can use FGCA for more specific pathways we're going to look at. You can use enricher to pull in different pathways as well too. For instance, I pulled in all the hallmarks for here to show you. But this to show you there's lots of different ways to do things. Lots of different ways to do things. I did want to show you one kind of thing that was cool. Enricher that I use a lot is this enricher. It's exactly the same package I just showed you. You can pull this package into R, but what you can do is just jump out if you want and just put your gene list here, right? And you hit it. And it basically just gives you a ton of stuff. Like here, you can look at every single one of these type of pathways, KEGG, Hallmark, Ractome. You can look at the Go ontologies here, your cellular component, molecular function, your cell types. So it's just a good, quick way to get an overview of what you might want to see, right? And then you can jump back in and do a little bit more specific analysis and get a better feel for what's actually there statistically, right? So yeah, so it's a great way of looking at the pathways. You can tell there's a lot of different ways to look at mutation pathways. So now that we've kind of come to the end here, I want to talk a little bit more about drug-gene interactions and basically how can you look at what genes could be actually functionally actionable, or what do we know so far about these genes as far as they match with different promising therapies. So obviously the ultimate goal is to match all the patients with your therapies, right? Based on your sensitivity, resistance, response. I mean, there's all different ways that mutations can be involved in helping us guide the right therapy interventions, right? So one quick way that I think is kind of cool here is you have this, Math Tools has something called drug interactions. Man, I told you, I have to do one dictionary mistake at a workshop. There's no way I missed that. I almost made it. So you basically just put in your pad math, drug interactions. It's basically going to tell you all the different genes that are associated with different drugs. So you have kind of a gene level of what different things can be drugged and what drugs can be used with that. But that's only at the gene level, right? We want to look at our variants. We have specific variants. So one thing you can do is use CIVIC. Instead of a broad lead scan, we want to go deeper into what is our mutation clinical actionable. And do we have more robust evidence for this? And CIVIC's a really cool way of doing this. The best thing you can do here is just every night that they fill out the new data. So whenever you run a new summary, it could be different. So you can just pull in that URL, write in R, and then read that TSV file, and you have the new file that is changed every time you run your new analysis, right? And here's kind of a glimpse of a lot of things that it is showing in there, a lot of different columns it has. Very, very, very robust. So what we're going to do is basically split. So right now we have the gene and the variant in the same column. So we want to split this into two columns to make it a little bit easier to read. That's all we did there. And then we want to do kind of a quick summary, and we kind of see the counts of some of these genes as we expect. Some of these are pretty high, just as expected. These are the genes not mutated. These are the genes that we have the most drug evidence for. So KRAS pretty high, T3 a little bit lower. So now we want to match and say actually we want to filter for drug actions specifically. So we want to look at just predictive. Remember, there's all those columns. We want to look at just things that are predictive, and just select out some common columns, and then we'll look at let's look at one gene. Like we'll pick KRAS and we'll say, okay, if you look at KRAS gene and you look at all the variants that are in our data, what therapies and significant can be attributed to those specific mutations, and what is the significance? So that's what we've done here. We've just chose evidence type that's predictive because it basically predicts response, right? Grabbed a few columns, filtered for KRAS, and then got the results. This is basically just a big data set. The answer is already there, right? We just need to filter for our stuff and match our variants, right? So that's why we pulled in, our original URL, right? We split the molecular profile. Which genes have the most evidence, and then now we're taking one specifically, and we're looking at KRAS only. So this is not our data yet. I'm sorry. So this is just looking at different KRAD. You can kind of see different mutations. What are the therapies? What's the significance of those therapies? Now let's join back to our data. So we're basically just taking our data, finding some of the important things like the variant classifications, the sample ID, the protein, and we're basically just joining it back in. And now we have our drug hits here, and here's kind of what we can look at. And we see like if we have KRAS and we have this mutation, this is the variant classification, tumor barcode, what are things that we could do with that, right? So it just kind of summarizes it all, what we're looking at. And so let's basically look at the answer to this. Now we have our answer, we have our full thing. So we're gonna take my drug hits and we're basically just gonna pull out these columns and take a look at it. And here's, for example, what we had. And here on top is that hypermutator, which is very interesting. And what's super interesting is ERBR2, there's only one mutation that I know of in that sample. And you can see that based on what's going on with that sample and that variant, we have different therapies and different significances on that. So basically you can pull up all your samples, your genes that are mutated in each sample, and your variants, and be able to see the therapies and significance of those therapies at the variant level. So I think that was pretty cool. And you can go a little further to change the names a little bit better and look closer at other things. But I'll let you kind of mess with that if you want. And this gives you a good idea. So we have 113 patients that have, out of 169, that actually have actual mutations. So that's pretty cool. And this kind of just shows you different ways of reporting some of these mutations and kind of a success story in pancreatic cancer with BRCA1 and 2 to be able to allow a drug to target PARP inhibitors. You can read a little about that if you want. It's kind of interesting. But that's kind of it for our basic thing. I'm going to go through the practice problems, but that kind of covers everything we have gone through so far, or we wanted to go through. We just want to kind of give us some reps here and try out a couple of new things. So the first thing we want to do is do a deep dive into CDKN2A. We know that's an important mutation, and we purposely left it out earlier. When we looked at TP3, KRAS, and SMOD4, we left this one out on purpose. So we wanted to check and see if a deeper dive into it. So CDKN2A, it regulates the cell cycle. It's frequently deleted in pancreatic cancer, and it leads to higher cell proliferation. If it's not there, it's a brake to keep that down. So that suppressor is gone, you get unlimited cell proliferation, and especially if you're messing with the cell cycle, you're in big trouble. So the first thing we're going to do is just calculate the mutation frequency. We already kind of did it above. You could just do the get sample info and filter for the gene name, but I'm just basically going to divide it myself here. And we're going to get here that 17%, 30 samples, have a mutation. This is the way you could have done it, the easy way, where the get gene summary. And comparing to other genes, it is in the top 4 of most mutated, so that's to be expected and good. And now let's look at the Kaplan-Meier survival curve. So basically, I basically just made a new survival data where I filtered for the CDKN2A mutated samples. I could have just added it to that last survival data and used it. But anyway, that's why we saw the CDK2NA on my other one, because we put that together that way. Remember how the survival data was different and missing the column? That's why it has it now. Because that's why I remade it that way, and now it has that column to call. Okay, so basically just identified those samples, I filtered by them, and I created a new mutation status. This is exactly the same thing I did earlier. There's no difference. I just made a survival data. It has the days, numbers. Remember, I interjoined my mutation status. It gives it the ones and the zeros and the actual genes. This is all we've done, exactly like before, and now we've done the same thing. Survival data, CDKN2A, SURFIT, exactly like before. And we're seeing it is significant. It's not super low, but it is significant. So in a bubble, if you have CDKN2A, the survival is lower. So that's interesting to see. And you kind of see just here kind of the difference in mutated versus wild type. And let's look at the Cox proportional hazard model now. So we're doing kind of a similar thing. We want to add TP53 first to that because we want to be able to put that into the model, and basically just cleaned it up a little bit. And we basically just have our 2 genes in there if they're mutated or wild type. And then we want to build our data. So we have our Cox data. I'll show you what that final Cox data looks like. So our final Cox data is similar to our survival data. You know, we have our 1s and 0s, our survival time, wild type, mutated, things like that. Just kind of got it all ready, very similar, almost exactly like before. And then now we're looking for all these things: CDK, TD3, age, stage simple. Now we know age, we know all these showed up before, so we wanted to say, if we throw in this 2, does it matter? And what we see is no, it doesn't. The only thing that came up is stage and T53, significant. If we look at the forest plot here, we see here that stage and TP53 are both significant. So if we throw in more things like KRAS, mod 4, we kind of lose a little bit of that. But we kind of already said we believe these are significant. Age was always in the middle. CDK2 and A, it's showing that if you have it, it's worse, but it's not significant, right? So really, when we look at everything together, we're really just seeing it's more KRAS, TP53, and stage that matter than anything, right? So let's see. Is this really an independent procrastinator? I mean, the hazard ratio is not too much. It's not significant. Not really seeing it. So we're going to say it's not a significant prognostic factor. And if you want to go by, also, you want to look at just stage. Let's do a Cox pH with just stage, and then let's so we just do that alone. This is interaction now. See the star interaction. So sometimes if you have a mutation in certain stages, it matters. So it may be that in stage 2, that if you have mutation there, then it has a bigger interactive effect. Okay, and we kind of are seeing that here. So for say the first one does not have it, but if you do an interaction, we're getting a significant value. So maybe there is an interaction. So let's actually plot the survival based on stage. So for example, this really, really red group is stage 2 and mutated, to where this brown one is stage 2 and wild type. So we're seeing stage is worse, which should be expected, and we're seeing stage plus CDK is worse. So both those interact to make it worse. Stage in itself is worse. And these two guys, you know, this is CDK and this is mutated. Actually, the mutated CDK is actually better than the other one. But their sample size is too small to actually matter. This guy's still alive, actually. Anyway, so you see what I'm saying? Stage is worse, obviously, but if you add the interaction of CDK to A, it's even worse. So that's pretty cool, I think. So now let's look a little bit more into the RAS pathway. So the RTK-RAS pathway is very, very important in pancreatic cancer, and it really drives proliferation, survival, metabolism, cell cycle problems, a lot of things. So what genes are all actually in the RAS pathway? So there are all these guys, right? And so we need to search for those and see how often we see those. I'm going to give a little more information about it if you want to learn, but let's just find these all. So we have 126 total among all our samples. And if you look here, KRAS is in 62.7%, but then we drop off like a freaking cliff, or even BRAF, the second one's only 1.2%. So we're barely seeing the other RAS mutations at all, which kind of makes sense because pancreatic cancer wants the RAS pathway to be disrupted, but it already has KRAS in almost all of its mutations. So when it's doing its thing and trying to figure out and scheming for how it's going to attack our body, it thinks, okay, I'm good. I'm good on RAS. I got KRAS mutated. That'll take care of me. So I don't need the rest. Where if RAS wasn't present, you might need some of the RAS mutated to help knock down that RAS pathway. But anyway, so let's kind of visualize these frequencies. Obviously, KRAS way ahead. Let's calculate the total pathway mutation frequency. So any genes, samples with any pathway mutation, now we're up to about 65%. So that's kind of interesting. So number of pathway genes mutated, 1. That's KRAS, right? 2, so 1.2% of the cohort has 2 of these genes mutated. And then we have that hypermutator that has a lot, right? And let's look at co-occurrence. That should be really interesting with these genes, right? We kind of already did that, but let's look a little again, and we'll see, just to think about it, is that KRAS is mutually exclusive with everything. If it's there, it doesn't need anything else. Where like some of these other guys, like ERBB2, ERBB3 want to be together. You know, a lot of these guys want to be together because you need, If you don't have KRAS, you might need multiple of the other ones to accomplish the same thing. That's kind of how I would look at it. So, and this kind of gives you just a manual calculation of everything. And then we do basically a Fisher's test to see if KRAS mutations are mutually exclusive with other pathway mutations, which we already kind of did that. But it'll show once again. Let's see what I was going to say. So what's interesting is it's saying that I'm not KRAS itself versus all the other ones. It's saying it's not mutually exclusive, but it really should be. So probably what's happening here is just there's so few of the other samples, it's just throwing out the test. See what I mean? This could be due to small numbers of not. So it's just too small to do that test. So the more robust test is not really needed because it's so obvious that they're exclusive, right? And I'll show you, you see the number of samples that have these. I mean, the number of samples that have both are only 2.2%. Basically saying KRAS and others. So 1.8% have others. These have both. So it's pretty obvious that they're exclusive. And let's just do an oncoplot. You'll really see it here. So we're just doing an oncoplot with the pathway with these RAS genes, right? You see KRAS is at about 60%. When you have KRAS, you don't really see the other ones at all. You only just see this one BRAF, one ARBB2. And when you don't have KRAS, you have this hypermutator and a couple more. But you're not really seeing much in general at all in this cancer. So that's kind of interesting. KRAS is really driving this. And look at the actual ones. This G12D area is really driving that strongly as well. So we really know that is really causing the problem. All right, so one last thing I want to go through is, now I've been teaching you the math, kind of that math tools data format. And I know I've showed you the pivot wide tidy format. I mean, pivot to long for the tidy format as well too. But I really want to stress that these functions these packages are just basically they're actually acting on just CSV file data, on Excel file data. It's just a matrix or a data table, right? So you can move these things around and transform the different things. If you have really a messy one that it's not in the right form, you can transfer it into the math form. Or you can transfer it into a different form. Or you can put dummy columns to make it fit the math tools. So there's all ways you can do to transfer the data into different ways to be able to use these packages, right? So I just kind of wanted to show you a way that we can still build oncoplots, lollyplots, blah, blah, those kind of things, starting with some messy data sets. So let me just show you a couple examples of how you can clean things. Obviously, things can be much dirtier. I've gotten a lot of dirty data sets the last year, and a lot of it you have to do unique code to each one, but let's just show you a couple simple ways you can do things. So basically, I just created this like what I call like a ground truth data set that I'm gonna kind of mess up and make messy, and we'll see if it works out. So I basically just built out a bunch of synthetic data and made it into a ground truth. And here's kind of the ground truth here. You have like different things like variant classes, the type of protein, the counts, things like that. And then now I'm going to basically make these into some new formats here. I won't go into it too much how I did it, but we'll kind of look at the result. I'll leave it to you to kind of see how I made these different ones. So our first messy format, what we're going to have two variants in one column. This is something that you often see where you see the colon separated by a semicolon. What do you do with this? How can I get this out to be able to put into a plot? So, and then messy format 2, lots of things going on here. The gene names are as columns here instead of rows. There's a lot of NA values. There's clinical data mixed in. Proteins are all over the place. So it's just kind of a mess. How can you put this into this kind of format that you can put into these to get these oncoplots, these lollipops? Now, obviously, you could also put this into a mutation matrix, right? Make all the zeros, NA zeros, make all the protein things 1. But for that, we don't want to do that today. Our third format has kind of mixed data types, and the columns are all weird names. You don't have the chromosome thing here, just the numbers. You have 2 weird columns here. You just got a bunch of junk going on, right? Inconsistent naming, protein change. Who writes that? So how can we clean this up? So let's look at the first one. We're the one that had the separating these semicolon variants. So once again, I'll kind of leave this to you, but I basically just made a new column called gene, and I basically just got the genes, all the string trim, as I want them to all be the same. Then I chose patient ID, and I put it to uppercase, to upper, make it all the same. And then I separated the variants by the separation, by semicolon. If it was a comma, you put a comma there. And then I rename protein, rename protein column to variants. Let's see. And then I just, and then I basically just plotted this out. And now it's all clean now. Look, now our patient IDs, our genes, our proteins, all our names match in the right formats. And they've pulled out like the doubler, that double Arita one, they pulled that one out and put it into a new column for us. So now, because the way it wants things done is just for each column, you have the patient ID pulling up each gene. All right. And then, so that's kind of how we want to clean that guy up. So that guy's cleaned up. Now let's see, part 2. So the 2 one, remember the 2 one that had all the NA values, mutation matrix, the weird thing. So we need to put this into a format like this. It's in the long format, right? This is the wide format. We want the long format, right? So patient IDs stay the same, but we need to put all this vertically, right? To be able to put in all the later stuff. So for this one, we're going to make 3 columns here: patient ID, age, and stage. And we're basically saying these are the columns that are the gene columns, not the clinical info. Then we're going to basically pivot from wide to long. We use pivot longer, and we have gene protein, and that's where we're going to put in all of our doublet things, our multiple things that were in that. We're basically putting our columns into rows, basically, right? And look what we've done. We're doing great. Now we have our patient ID column, we have our clinical data, and now we have all of our genes. Now instead of that wide format, now they're in the long format and split up. All the NAs are out. And you see, for instance, for patient number 1, you have these 3 types of mutations. So that's much, much cleaner than what we saw earlier, where patient number 1 had all the NAs, nonsense, extra rows, things like that. And now we can be able to put that into our next thing. And let's do our final one is kind of cleaning that the weird one. So first of all, we kind of sliced out that weird top rows, and then we kind of we basically just redid all the names. These are the names on the left that we changed those names to, and then we basically fixed the chromosome annotation by adding the CRH back, removed any NAs, and here we go. It looks pretty nice, right? So now we get the long format, our genes, proteins. We had to fix the chromosome thing. All of our labels are the same and correct and look nice. So that looks really good here. So now that we've kind of tidied our data and built our data frames, let's kind of make these into a math file and try to put these back. Now, we don't have to do this. We could use those for other things too, but I'm just going to show you an example of how you can put it back into a math file. So we're going to start this with the ground truth, make this into a math-ready file. So we're basically just saying we already have this variable on the right. We need to rename it to the left stuff for the math file to know. So all I've done is just renamed everything and then put it back in, right? And then for the tidy ones, so the tidy 3 does not have variant classification. So I'm basically inferring that from the protein change. So basically the same thing. I'm just setting the new rows, setting the new name, sorry, and then just predicting these different protein ones. And then for tidy 1 and tidy 2 is basically the same thing. We just basically fix the variant classification, and then we put in our rows. They're a little bit different. We have some dummy rows we need to put in, just so it'll work here, because we don't have some of the information. So this is just showing you how you could put some dummy rows in, because it's going to look for these. It won't care what they are, because it's just looking for certain things. So we basically run these 2 functions, and we have our different ready files here, and then we have our math 1, 2, 3 to get our 3 different new math objects. And if we look at one of these math objects, synth math tidy, basically going to get what you expect there with all your data coming out in your first guy, right? just like expected. And then basically we can use any of these ready files now for all of our downstream stuff. And just for fun, we'll do it with the simp map, the first one, the map ready one, and we'll just run that through. You can make an oncoplot. I can use the simp map the same way to do my ggplot, my lollyplot. I can do a clustered heat map of these mutational profiles. I'm basically just, I made a math ready file. I can just use that math file now, just like I would use it for anything else, to do my clustering, to do my pathway enrichment. It's nothing different than we haven't done before. Exactly the same things, except I'm being able to use that math file, right? That math ready file, right? So my original simp math. What was my first one? Yeah, simp math. So this, just doing the same thing. Put it in the oncoplot one. I'm basically putting it in the lollipop one. I'm basically just doing a clustered heat map with heat map. See a simp matrix, binary clustering word 2. Pathway enrichment. I basically just grabbed the genes out of there. Did our normal pathway analysis. Since it's synthetic data, that's why you're getting a weird result here. But I just want to show you the pipeline and the code. Now remember, you can put whatever cancer type you want in this whole data, in this whole workshop. You can put, whatever type of messy data in here and make it into a math file. It'll work. That's the point. I want you to have these data pipelines to be able to build off of. And then I just kind of summarized some stuff here for you. Gave you some key publications, some online resources you can look at to learn a lot more about this stuff, and some of the packages we use. The survival packages, cluster profiler, FGCA. I'd encourage you to go back through that one file, the one part of the code block where I have all the libraries, and basically just drop those into ChatGPT and ask to learn more about them, and give me some documentation links to some of them, if the ones you care about the most. Really getting the idea of finding things that you want to learn and throwing them into Claude or whatever, and having it run up some summaries for you to learn more. It's a really, really quick way to learn. So we're finished our workshop 2 here, and we'll kind of did a good job of kind of connecting this back to the clinical data, looking at more statistical associations and mutations. And then next week in our next workshop, our final DNA workshop, we're going to be looking into mutation signatures. We're discovering the biological processes that actually cause these mutations. We're gonna use NMF for our own de novo signature extraction, try to find some new signals. We're going to use some cool single cell data to try to understand and learn some things about tumor evolution, phylogenetics. We're going to go a little bit farther with the DNDS analysis for driver genes, and then we're going to use a machine learning model basically to predict cancer types from mutation profiles. So that should be really cool, and I look forward to seeing you guys for that 3rd workshop. Thank you.